Welcome back to another video done by me, Julio Cesar Benitez Jr. Today I am going to be discussing about the adult midgut in insects, blood-sucking insects to be exact. Introductory. What is a midgut? This might be the first thing we need to cover in order to understand the whole chapter. The main idea of this function is the ability to digest and absorb blood meals. It is also where the parasites and viruses enter the vector for subsequent transfer to a vertebrate host. Blood meals can be categorized as either a batch or continuous. Batch occurs when digestion proceeds simultaneously. This basically means that the whole process is done in one sitting. The other category, continuous, is when the blood meal is taken in sections. It is first held in the anterior midgut, where there is no digestion occurring. Then portions go through the middle and posterior midgut, where digestion and absorption occur. Blood meals are normally separated from the midgut epithelial, where digestion and absorption happen, by an extracellular layer known as the peritrophic membrane, or PM. This membrane is thought to protect the insect from outside intruders that may have slipped inside, abrasions, poison, and ease the process of digestion. The majority of the blood meal is made up of proteins. The rest is considered as water weight. 20% of the blood meal consists of protein. Lipids and carbohydrates are almost insignificant in the blood meal. Amino acids from the blood meal protein synthesizes lipids and carbohydrates, stores involved in egg production. The use of amino acids is an important source of energy for egg production overall. The size of blood meal varies from species to species. Factors may even come into play if we involve temperature, age, mating, etc. The general blood meal size is twice to 10 times the body weight of the insect. This can impair mobility and stress to the gut. Most insect species, though, have adapted mobility by the process of diuresis, basically urine, and handle stress very well by having modified parts or the ability of elasticity. The time required for blood digestion may vary from 4 hours to 48 hours or more than 300 hours. This is equivalent to basically 12.5 days. The last section of the introduction is lysis of erythrocytes. The whole concept of lysis of the red blood cells is to gain the protein that is within the erythrocyte. There are many examples like the use of the proventriculus, which is used to physically break down the cells. The proventriculus can be seen as a pathway with teeth in order to shred the cells, or you may think of a shredder if that is easier to imagine. The other examples are non-physical, which are the use of cibarial armature and chemical hemolysis. This can be sometimes secreted by the saliva of the insect. The most common form of lysis is chemohemolysis that is produced in the midgut. Structure. The insect gut has three sections. These are the foregut, the midgut, and the hindgut. The foregut's job is to get the blood meal from a potential host into the vector. It uses pumps in order to draw blood and may store them inside in a section called the crop. Some have a crop while others may go directly to the midgut. The second and the main chapter's concept is the midgut. It can be divided into 2 or 3 sections: anterior, middle, and posterior midgut. Salivary gland secretions are used to make the blood easier to pass and digest in the midgut. These secretions can be found close to the mouth parts of the insect. The last part of the midgut is the Malpighian tubules. These tubules are considered the insect's kidney, in which it filters its own fluid, hemolymph. This is a fluid equivalent to blood in most invertebrates. The last section of the gut is the hindgut. The hindgut reabsorbs ions that may have passed by and creates excrement known as frass. The midgut has 3 or 4 different types of cells. The majority of the cells are named digestive epithelial cells, which produce and secrete digestive enzymes to which have prominent secretory vesicles. The second type is named absorptive cells. These cells are difficult to compare to digestive cells due to their likeliness in structure. The third type is named endocrine cells. It is said that these endocrine cells produce several peptides, but there is no information in what these peptides do. More research needs to be made in this area of entomology. The last and fourth type of cell is named regenerative cells. These cells are undifferentiated cells which differentiate to replace midgut cells that are sloughed off. Hemiptera. The hemiptera or true bug's midgut is divided into two parts: the anterior, which is the stomach, and the posterior, which is the intestine. The interior portion is not involved in protein digestion and absorption. Instead, this is the site where food is kept as storage. Water is excreted from the hemolymph through the kidney, and the ions are kept as spheroids. These spheroids dissolve and are released back into the hemolymph. Carbohydrate digestion also occurs in the anterior midgut. The posterior midgut is the site of protease synthesis and secretion, protein digestion, and amino acid absorption. The posterior midgut is the major site for nutrient absorption. Diptera. Culicidae, or the mosquito, has two regions of the midgut. These are the anterior and posterior. The anterior midgut does not involve blood digestion. The posterior midgut is where digestion and absorption take place. All four types of cells are located within the vector. Cell division does not occur in adults. Maturity of the midgut appears 3 days after eclosion. This means the time when the organism is creeping out of the egg stage or cocoon stage. During the interim, the vector may feed on nectar or sugar water until they are able to take in a blood meal. Psychodidae's midgut act very much the same way as Culicidae. The Simulidae, or the black fly, have not been studied. The only thing that they have been studied on is the formation of the peritrophic membrane. Mucidae, which is typically the housefly but the chapter covers the stable fly instead, have three midgut regions. These are the anterior, central, and posterior. The anterior midgut is the site of stored blood meals. It is also the site of water and ion reabsorption. The central midgut is the site of digestive enzyme synthesis and secretion. The posterior midgut is the site of absorption of digestion products. The Glossinidae, or the tsetse fly, is similar to the Mucidae. The major difference is the presence of mycotomes between the anterior and central midgut, whose cells contain endosymbiotic bacteria. The last diptera is the Siphonaptera, or the flea. The flea is very different from the other insects that we covered. This insect has no regional specializations of the midgut. Instead, the midgut expands or undergoes distension during a blood meal. Salivary glands of blood-sucking insects do not produce digestive enzymes. Instead, they are produced in the midgut. The midgut has a specialized system for efficient digestion of proteins. This is where classifications of proteases come into play. There are many types of proteases depending on their substrate cleavage site, active site, and substrate specificity. There are two main classes of proteases. These are exoproteases, which release amino acids from the termini of proteins, and endoproteases, which cleave proteins at the carboxyl site of specific amino acid residue. There are also catalytic agents that can change the protease. There are metalloproteases, which contain a metal ion, serine, cysteine, and aspartic proteases, which are specific to the amino acid being used. Overall, the completion of digestion of proteins require both exoproteases and endoproteases. Proteases are really good in cooperating with one another. In all blood-sucking insects, there is an increase in digestive enzyme activity in the midgut following a blood meal. Digestive proteases also play a role in the activation of disease agents in the midgut of blood-sucking insects. Below you will see classifications of digestive proteases that may be found in the midguts of blood-sucking insects. Carbohydrate digestion and lipid digestion. Carbohydrates are used as fuel or energy for flight in many vector insects. This is very important in means of transportation and acquiring blood meals from hosts. Blood meals, as discussed before, have about 20% protein and 80% water. Even though this closes off at 100%, somewhere there contains other macromolecules but low in numbers. Blood meals have a very small amount of carbohydrates compared to proteins. This forces the vector to feed on plants for their nectar or other plant fluids that can be useful. These sugars are then stored in the crop of dipterans, then transferred to the midgut for digestion and absorption. Other vectors may not have a crop in their system and may just go directly to the interior midgut. The next section is lipid digestion. Lipids derive from the erythrocyte cell membranes. Small amounts are found in blood meals, just like carbohydrates. It uses amino acids that is taken from the blood meal to produce lipids for storage in eggs. There is very little knowledge about lipids in blood-sucking insects. The role of phospholipases in the lysis of erythrocytes and subsequent release of cytoplasmic protein. Basically, it can mean that it could play a role in disrupting the cell membranes. Amino acid absorption and carbohydrate absorption. Amino acids derive from blood meals and it is used to support egg production. The process starts with the blood meal being degraded into amino acids in the lumen of the gut. It then gets absorbed by the midgut epithelial cells and then transferred to the hemolymph. This will then get taken up by the fat body and other tissues. There are two types of transportation of amino acids. The first is the apical membrane, which faces the midgut lumen, and the second is the basal membrane, which faces the hemolymph. Carbohydrate absorption is very unique for insects in that it lacks active transportation systems for glucose uptake. Instead, they use unique mechanisms to create a concentration gradient in glucose between the midgut lumen and the hemolymph that ensures efficient passive transport. Lipid absorption and transportation. There is a major problem with lipid absorption in that lipids are hydrophobic. The lipids need to pass through the aqueous lumen to the hydrophobic cell membrane and into the aqueous cytoplasm. There is very little to no knowledge in how lipid absorption works. Insects require cholesterol and several species accumulate fat droplets in the midgut during blood meal digestion. This is a good way of using the lipid while digesting. In terms of transportation, lipids are transported in a lipoprotein complex called the lipoffurin in the hemolymph. There are many kinds of transportation types that use the lipoffurin. More research indeed needs to be made in the absorption and transportation of lipids. Thank you for watching my video, and I hope you all learned something new. Have a nice day, guys.